Okay, raise your hand if you're not participating in this right now. Okay, just making sure because I don't want to waste all of our time. Okay. So this is an easy one. Who's going to get called on for the easy one? Netta. Sorry. Your name just jumped out at me.
All right.
So I'm going to actually kind of keep track here. Do I have paper? I'm going to write on the board. It's going to be very exciting. Okay.
So the first thing I did was I drew out the 4 different phases.
Okay, so you said there are 4 phases of the cell cycle. And you did probably something like this.
Yeah. So I had G1 that went to S, that went to G2, that went to N.
Okay.
And then between G1, S, G2, I kind of did like a thingy that encompassed all of it and said interface. Or maybe that was wrong.
So basically you went like this, and you
Mitosis.
and Emma's mitosis. Okay.
Then I just wrote, there are 2 growth phases named G1 and G2. S phase is a period when the chromosomes duplicate. Mitosis has several steps, but is highly regulated.
Okay, so G1 and G2 you call growth phases.
Right.
What else is happening during G1 and G2? So growth and what else?
I guess organelle.
So that is growth, organelle duplication. What else? Sensing the environment,
Okay, yeah.
making sure the environment is right.
Well, I had another part. I had between each phase there are checkpoints performed by the cell in order to be sure that all conditions are.
Okay, so you mentioned, okay, so there's also G0, right? What's G0?
That's when the cell rests.
Yeah, the cell's a rest. Usually it's when something can't be repaired, the cell will go into G0. Checkpoints. So you said something about checkpoints. What are checkpoints?
There are ways for the cell to check that everything is basically in working order.
Okay. Mitotic spindle has attached to the chromosomes, right? So that's the metaphase to anaphase, right? So okay. Let's see. And we'll draw that into our picture. So that's somewhere over here, metaphase to anaphase, and we're going that way. So let's say that might be metaphase and that'd be anaphase. So G1S. What's going on there?
It checks to see if the DNA is properly duplicated.
Wait, G1S? Try again.
Oh, no, I lied. That's SG2. G1 is to make sure that the cell is properly grown and that the environment is proper.
Right, so environment. Are we ready to start? And so what's this other one? And what are we checking for? Make sure DNA is fully replicated. It all kind of makes sense. It all kind of makes sense. So as I was walking through, someone asked me a question where they were kind of confused, and I'm sorry to use you as an example, but everyone can learn. And they're asking me about, I think, what, like, maybe this checkpoint, metaphase, anaphase, happening up here. And I said to them, wait a minute, stop and think logically about how this all happens. It's a cycle, right? We grow, we replicate our DNA, we give it to our daughter cells. That's cell cycle, right? And so you don't want to talk about your metaphase to anaphase checkpoint if you haven't done DNA replication yet, right? And so be careful that you don't get bogged down with memorizing this as a list, because then you will start being like, oh, during, M phase, we replicate our DNA. Well, M stands for mitosis, so we should be segregating or whatever. And so I want you guys to really, this whole unit is extremely logical. Really, really so the last one with the cytoskeleton, there are all these proteins to remember. Here there'll be some proteins to remember, but all the things that are happening, if you stop for a minute and think about it, it all makes sense, right? You don't want to go from metaphase to anaphase if your mitotic spindle hasn't attached your chromosome, right? Because anaphase is when you segregate them. What happens when your mitotic spindle is not attached to your chromosomes and you segregate? What's going to happen to those daughter cells? You won't have equal numbers of chromosomes in your cells. Is that good? No. Is that a good reason to stop things? Right. Okay. So what other things that did people think about? Yes. P53. We'll learn about P53. What class of proteins, aside from tumor suppressor, would you put P53 in? What kind of protein is it? Is it a kinase? Is it a transcription factor? Is it a cytoskeletal protein? What is it? Does anyone know? It's a transcription factor. Okay. So we will learn about P53. Oh yes, we will. What else? Yes.
CDKs.
CDKs. What does CDK stand for? Cyclin dependent kinase. It regulates cell cycle. How? What do cyclin dependent kinases do to other proteins? They phosphorylate them. Right, so those proteins, so then you can think to yourself, okay, so if we're phosphorylating other proteins at certain points in the cell cycle, this regulates cell cycle, we must be turning on and off the appropriate proteins to help carry out that phase of the cell cycle. That's what CDKs do. So what do they, what protein regulates cyclin dependent kinases? Huh? Well, go a little bit more deeper before kinases. The kinases do help regulate CDKs, yep. Anyone? So it's called cyclin-dependent kinases because cyclins need to bind to them so they can be active. Yeah. CDK. Okay. What else? What else did people think of? Nothing? Yes?
The APC.
APC. What was APC?
That's the antigen presenting
Oh, you are thinking about two things at the same time. There are APCs, which are antigen presenting cells, and they are what are you thinking immunology? No, maybe. But so APCs are antigen presenting cells, but in cell cycle, do you hate how people use the same letters for different things?
I was thinking more back to
Yep, anaphase promoting complex. Yeah. Yep, but they're the same. They're both APCs, right? So, and we're going to learn about that. What do you guys think the anaphase promoting complex does? Promotes the anaphase. Right. What kind of protein complex is it? Does anyone know? Yes, it promotes anaphase, but by doing what? What class of proteins would you put this complex into? Hmm? It's ubiquitin ligase complex. So this it ubiquitinates things to promote anaphase. So we'll learn about that. Okay, I saw your hand up.
We1 and CT25.
We1. What was We1?
I think that's the one that inhibits the cyclin-dependent kinase.
Right, it's a kinase, right? So it inhibits CDKs, right? And then you said CDC 25. What is that?
That's a dephosphorylate.
Right, so it's a phosphatase. So these two kind of go together, right? We wanted CDC 25 because one turns one thing off and the other thing turns it back on. And we will learn about that. Okay, anything else people have? That's good. People are coming in with a good amount of stuff. As long as you have this, if you're coming into this class with at least this, we're in good shape. I do not expect everyone should know this. Some people have seen it before, and that's great if you retained it. Hopefully after your time done with you, you'll retain it even more. Okay, good. Let's start. Okay, so quickly. Business. Exam 3, I promise it'll be up during or after the holidays, somewhere around then. Things have been extremely busy, so apologies there. So questions that you all have right now, because this is the time of the semester where everyone starts calculating their grade rather than studying. I want you to stop that. Use the time to study. Do not sit around calculating your grade and guessing what you need to get, because everyone knows if you get 100, your grade will be better. But if you're not studying, you won't get 100. So it's not rocket science, and you need to get a higher grade to get a higher grade. We all know that. Don't waste your time calculating. Do I need to take the final exam? If you look at your average right now on Blackboard, forget taking things out, and you're happy with what your average is, then don't take the final exam. Do not come here because you feel bad, or else I will be mad at you. Okay? Come here only because you must, or because you love me. If you're on the cusp and you really need to know if dropping this and dropping that is going to do it, then email me, and I'll try to figure it out for you. But do not waste your time doing math, because I could easily press a few buttons and tell you exactly where things are. Is the final exam cumulative? Yes and no. It's going to be everything from the last exam onwards in depth, but there will be things where I'm going to say, signaling through this affects the cytoskeletons for the cell cycle, where you're going to have to put things together. So just make sure you're integrating everything throughout this class. Do we have a quiz this week? It's Thanksgiving. Yes, sorry. I'd like to not give you one, but you want one because you want to be able to drop some out. So that's why we're doing it. You cannot take it. It's always optional to take a quiz. No, you don't ever have to take them. Same with exams. But if you want to drop this one, just don't take it. That's your choice. Do we have recitation this week? No, we do not. No recitation today. We do have recitation next week, so make sure you look to see what paper is going to be assigned.
Is that already up?
Is it already up? No, it is not. I will get it up hopefully today or tomorrow. Okay, we did that. Yeah.
For the final exam, do we get to do the multiple choice questions for Lisa?
Yes. Final exam, you will get to do multiple choice questions. Final exam will be in this room on the final exam schedule on paper. There will be no oral exam option, just because this is all dictated by the university that I have to do the final exam at a certain time in a certain place. So far, the reason I have these up here is because I don't want you to forget that these are things we learned that have not been on any exams yet. So this is the stuff from the cytoskeleton unit that will end up on the final exam. Okay, cell cycle. So the cell cycle, basically all it's doing is controlling the copying of the genome and organelles and distribution to the daughter cells. That's it. You wouldn't want to distribute your genome before you copied it, so that's why it goes S phase to then M phase. It's all very logical. Oh dear, we're gonna ignore that the text is looking messed up right there. Right, so you'll have cell growth, cell division, chromosome segregation, cell division, cell growth. That's the cycle over and over again. And so you can think of the major elements of the cycle as S and M phase, right? Chromosome duplication, distribution to daughter. The DNA is replicated towards the end of interphase, and if all goes well, the cell will progress to M phase, where the duplicated chromosomes are segregated as a result of mitosis. So interphase is everything but mitosis. I've said this a few times, but people always think interphase is part of mitosis. It is not. Interphase is everything from cytokinesis all the way to prophase. Okay? So this is your classical cell cycle figure that you've seen a gazillion times probably during your education here at Towson or elsewhere, right? And so you're just seeing that you've got your G1, S, G2, M. And we talked just now about the purpose of the gap phases, right? That's mostly sensing the environment and growth and making sure the previous phases are properly. We've got G0 that occurs before S phase sometimes. It depends on the conditions that the cells are in and the cell type. What cells are always in G0? What else? Eggs. Right? Aren't they in G0 in cell fertilization? Eggs are eggs dividing all the time? Come on, guys. Sperm dividing all, are they just dividing all the time? Well, do sperm divide? Don't they get made by the testes? I don't know. I'm not a physiology person, but as far as I know, they get made and then they're there. They're there. Exit. Do that there. Exit. This is not health class. You know, I have little kids, so I have these really interesting conversations with them about all this stuff. And so, and I'm kind of like, the more they know right now, the better, because it's not awkward yet, right? From like 10, 12, 13, they're going to be like, Mom, don't talk to me. Anyway, sorry, just think about sperm. Okay, checkpoints. We spoke about checkpoints just now. So cell cycle, it's tightly regulated, right? And so it's controlled by all these different proteins, and their whole goal is to either allow the cell to progress or not, right? And the cell shouldn't progress to the next phase of the cycle unless the previous phase was completed appropriately, right? And so these checkpoints we spoke about, right? Here's your start checkpoint, is the environment favorable? Your G2M checkpoint, is the DNA replicated, right? So what there's DNA damage that prevents completion of DNA replication. So you do not want to segregate your chromosomes if DNA replication hasn't been completed, right? That seems like a bad choice. Metaphase to anaphase transition. You don't want to segregate your chromosomes if the mitotic spindle grabs onto this chromosome. So I keep saying cell cycle is highly regulated and it's controlled by many different proteins. Why does this matter? Why is this so important? Other than the fact that Dr. Early was telling you. Yes? If it's out of whack, you get cancer. Exactly. That's the scientific way of saying it. Yes. Right, and you can get other diseases if you don't have equal splitting of chromosomes, if you have unequal chromosomes. Yeah. So we're going to spend a little bit of time going through how cell biologists study cell cycle regulation. Who is signed up for the cell lab next semester? Anyone? Okay. The rest of you actually, those of you who have a lab requirement, who has to be filled and are trying to get into more bio lab for 10, don't forget there's a cell bio lab that's starting up next semester. And so you might get to learn a little bit, I don't know, some of the stuff. Am I teaching it? No, but I will be helping. Dr. Snyder's teaching it, and there will probably be something in the fluorescence that will be really helpful. You'll get to see my smiling face. Okay, so first let's talk about model organisms. As you may have noticed, cell biology research is done at least a lot in frogs, right, in Drosophila. So that's because a lot of this cell cycle machinery is highly conserved. And so it's easier to study it in model organisms, such as yeast, animal embryos, and then of course cultured mammalian cells. That's not really a model system. So this is showing yeast. That is a frog. Everyone, I hope y'all can this. This is a frog. Right there. What kind of frog is that? Xenophil. Yeah. I'm like, don't ask me what their common name is. And these are cultured cells. I think they're HeLa cells actually. Oh yes, I love you, PowerPoint. Though my extra memory has been ordered, and I believe it might be sitting upstairs. So by the end of the semester, I have no memory. Saccharomyces pombe and Saccharomyces cerevisiae. These are a commonly used model system for cell cycle regulation because they're very easily genetically manipulated, and you can visualize them easily. You can tell what's going on. So here's fission yeast, and they split through fission, and budding yeast, they form these buds. What's budding yeast? You can visualize. You can see if there's a mutation. You can create these mutants, and you can see where did they stop in cell cycle, what proteins are involved. So one of the ways that this is done is through conditional mutants. And one type of conditional mutant is called a temperature-sensitive mutant. And these are mutants that basically the way it works is the mutation is in a gene, and it's not such a horrible mutation as a normal growing temperature. So the protein is folded normally, and the cell can function normally, and you can propagate the yeast. But if you raise the temperature a little, it causes that protein to slightly misfold, and then you can actually study the phenotype of that mutant. So usually people get really confused about temperature-sensitive mutants. But just the key is that you cannot study something that arrests the cell cycle, because how can you grow the organism if it arrests the cycle, right? So growth can't happen if cell cycle has arrested. And so that's why they have these conditional mutants. And so the example I'm showing here is showing here's your G1S, G2M, that's your cell cycle, and you've got these cells growing in culture, and they're all at different phases of the cell cycle because they're not usually synchronized, right? You might have one called F, another called M, and so that's just how they're going. And then when you increase the temperature in some mutants, that will cause one gene, which you're studying, to be mutated or misfolded or not function properly. And it'll have an effect on cell cycle, cause cell cycle arrest. And so the cells will continue cycling until they hit that block. And in this case, the block is somewhere at the G1 to S temporal. Okay? And that's how you study them. So way back in the day when they were studying all these CDC proteins, the way they the way they characterized them was through these types of conditional mutants, and they would have your permissive temperature where the cells were normal, and then they would shift the temperature to the high temperature, and then that protein that was mutated would stop functioning, and they would look and see what point of the cell cycle have these cells stop growing, and that would be able to help them to begin to characterize the function of these proteins. Right, so I guess that's always a possibility. Ideally, with a temperature, the question was, what if you increase the temperature and affect more than one protein? But you're not heat shocking. You're not like raising the temperature to a point where all the proteins are going to misfold. It's the kind of mutation that's so subtle that you just shift it from 37 to 42, and it's going to cause, that protein to stop working. Now, there's always a chance, because I know sometimes they have these engineered mutations and sometimes it's randomly generated mutations. And so there's always a chance that there could be multiple mutations, and I guess in those situations you wouldn't study that. You would not use that strain to study anything. Yeah?
Does it, when you do the temperature shift, does it have to be like misfolded because you just not know the protein entirely in it for the study?
So your question is, does it have to be a temperature sensitive mutant, or can you just knock it out?
Well, could you knock it out but using a temperature controlled knockout? Or does it have to be like this mutating so that it doesn't fold off?
Well, so my example of how that temperature sensitive is with it not folding properly may or may not be completely accurate. It's the easiest way to understand it. Sometimes you'll have temperature sensitive mutants. There are other kinds of conditional mutants. Sometimes you need a drug. There are different ways to do it. If you knock out a gene, that means you're removing the DNA from the genome. And so then that gene's no longer there, and then you can't propagate that cell line, that mutant strain. You know what I'm saying? Yep. Yep.
I'm just confused how to stop it at a certain point of the cell cycle, not just
So you're wondering how come they're not going to be zero? So this is like, so think of it this way. I like that because you would think if you can't, so G0 is usually reserved for when something cannot be repaired, right? But in this case, you're going to go once around stuff, right? So you have this culture of cells growing, and it's not like there's DNA damage. Right, DNA damage can be repaired, G0, stop, wait. But in this case, these are specific genes that you don't know anything about, you're trying to study. And if you, let's say it's CDK that's important for going from G1 to F, it's a mutated gene. Well, then it's going to stop at G1 to F. It's not like you're causing DNA damage. You know what I'm saying? Yeah. Yeah. So think of it this way. If you have a normally folded protein, should it work normally? Yeah. If you have a misfolded protein, should it work normally? No. And so when you shift that temperature, so at normal temperature, this is the permissive temperature, and it's not really low temperature, it's just normal temperature. Think of it that way. The protein's folded normally, and it doesn't show anything, that's a control. But when you do the restricted temperature, which is the high temperature, you cause that misfolding to happen, then whatever phenotype that mutant's going to have, you can see it if it's in cell cycle. Because you're specifically looking at cell cycle. Right. Right. So you might have 10 different strains of yeast. All of them are temperature sensitive mutants, but they're all going to have a different gene that's been mutated, and all of them, when they're at that permissive temperature, which is the normal growing temperature, they're going to grow normally. But when you shift it up, that one protein is going to mutate, and it's going to have an effect on the cell cycle, which, depending on what you're looking at, you can start to identify and understand. Okay. So if you look at this example, okay. So this is temperature-sensitive mutant of CDC15. And this is how all the CDC proteins were identified. So you can see in A, you've got your permissive temperature, so you've got your wild type phenotype. And in B, you've got your restricted temperature. So you've shifted the temperature up, so your CDC15 is no longer working. Okay? And so you're going to look at them and decide what's wrong. So I want you guys to take a minute and look at them and tell me, what do you think is going on? How do these look different between when CDC15 works in A and when it doesn't work in B? So just take a minute and you can talk to your friends if you want. Okay, guys. So, what did you guys see? I heard from this side of the room a little bit. Sorry, not on this side. What about this side of the room? Yeah.
Would it be putting a recipe in here and like that it's starting to separate or not actually separated yet?
Right. So something is going on here, right? Every single one is stuck in the same phase. Whereas here you're at different phases, right? Okay. So something involved with cytokinesis. So what do you think might be involved with cytokinesis? Anyone? Yeah. Contractile ring formation. Right. Right. So it turns out CDC15, and I guess you all could have sat there and Googled it if you wanted to. Without me cheating and not using your brain. But yes, contractile ring formation is regulated, right? Obviously. Who thinks contractile rings just form randomly? That makes no sense at all. Obviously it's regulated. It only should be happening basically around just prior to cytokinesis, right? And so then you've got to think to yourself, well, what controls that? Some kind of kinase, probably, right? So exactly. So contractile ring formation is being affected. These cells are being arrested at the end of mitosis prior to cytokinesis. These are all things. So this is, if you were in the lab studying this, this is exactly how it would work. You would look wild type, mutant, what's the difference? What do I know about this? And then you would start to be like, okay, and cytokinesis, something with contractile information. Let me look and see in the literature what's out there in the literature that's known, and what tests can I run to kind of confirm my hypothesis. Good. Okay, so that's yeast. Also, we can use early embryos of Xenopus, Spicula, and Drosophila, and you can use these for studying cell cycle because they're very simple cell cycle and they're very large, so you can easily look at them. And you can look at growth of the embryo. There's a lot you can look at there. So there are whole labs that study cell cycle using these kinds of models. So they're never working with mammalian cell culture. And I always tell people, go into these labs because the tools are amazing of what you can do. There are mutants for everything. But it's also a really, really competitive field. So, pros and cons. More tools equals more competitive. So the cytoplasm isolated from a xenophus egg can be combined with sperm nuclei, and then that could be used to study DNA replication and mitosis in a cell-free system. You can use immortalized cell lines. What are immortalized cell lines? Cell lines that always divide. What usually happens to them that aren't immortalized cell lines usually cancer cells? Often they are. They're transformed cells, so they're either cancer cell lines or they're cells with some viral genes in them, like HeLa cells. Who's heard of HeLa cells? Now everyone has, right? Because of the immortalized Henrietta Lacks. So yes, HeLa cells have HPV 67 in them, and that helps them to be constantly dividing. And so because they can grow indefinitely, you can study things in them. If I just take cells out of your skin right now, you'll get a limited number of divisions, and then the cells stop dividing. They become senescent. So if you're studying cell cycle using mammalian cells, you can do a whole bunch of different things. Also with yeast, you can do a lot of these same things. You can directly visualize them. So that's what we just did with the yeast. All we did was just look at them. And you could see these were dividing, right? Cells in mitosis are rounded. You can see cytokinesis easily. You can also visualize with DNA stains, right? So DAPI, which we've seen over and over again to stain DNA, or specific antibodies against specific proteins. So in this picture right here, what are we staining for? Who wants to tell me? Who wants to guess? Yes. So the blue is DNA, and what's the other one, the green one, is antibodies against what protein? Against tubulin, right? Because these are your microtubules. We just learned about that. And the reason you can tell is because you've got your mitotic spindle here. So what phase of cell cycle is this cell in? Metaphase, right? What phase of cell cycle is this one in? Prophase, probably, because you haven't duplicated your cytosome yet. What phase of cell cycle is this one in? Interphase. So you guys are ready. You guys are ready to just jump right into research. But you know this stuff. People think these are so hard, but you know it. Why is there a cytoskeleton? For what? Those are your microtubules. No, they're not. This is okay. With immunofluorescence, you can only see what you stain for. Right? So then no one is stained for a membrane. So you can't see the membrane. Right? So what you do when you do immunofluorescence is you actually treat the cells with a detergent that's going to poke lots of holes in the membrane, and that allows the antibodies to get inside and stain the protein on the inside. But before you did all that, you fix them with something like methanol or paraformaldehyde, so it kind of keeps everything where it is. So it's kind of like freeze at the time, poke holes, add antibodies or binds to whatever, add something secondary antibody with color, boom. Yeah. Yeah. So the question I just got is, what can you stain for with immunofluorescence? Anything that there's an antibody for. Yeah, so you name the protein, you can stain for it, yes. Because I know that this is the mitotic spindle. because it looks like it. I mean, I know because I picked this image and I said it looks tubulin, and if you were doing the experiment, you would choose to stain with what you wanted to stain for. But just coming off of this cytoskeleton unit, I would expect that you should be able to recognize microtubules. And so that's why I asked you guys. If I threw out any immunofluorescence image, I would not expect you to know what it is because you can stain for anything in the world. You could stain for ND10. You don't know what ND10 is, so why would I ask you, right? But with microtubules, I know that you guys know that your interface microtubules look like this, and your mitotic microtubules look like that, just because we just ordered it. Any other questions? We have a really wonderful immunofluorescence microscope here. And so every now and then I use it, but people do use it, and so this kind of stuff is going on. It's pretty cool. But there are also ways, if you go, we have a thing with Loyola, they've got a confocal microscope, and so that will take, but what basically that does is it'll focus, like slowly, automatically focus up as the pictures are being taken with a laser, and then reconstruct it so you can get like a 3D image of the cell. It's very cool. Very cool. Anywho. Okay. So you can look at cells. You can stain for certain things. An example of something else you can do, which is involving immunofluorescence, is you can use BRDU. And so BRDU is a thymidine analog. What's thymidine? A base, right? What are bases? Okay, they're part of your DNA. I was reading something in the Atlantic or the newspaper yesterday, and it was the saddest thing ever. It was like, it said something about how you can make any protein you want. You just put it, no, you can make any DNA sequence you want. You just put in the right amino acids. And I was like, in the newspaper, or somewhere, society. BRDU is a thymidine analog. This is a base. When are thymidine analogs incorporated into the DNA? During what phase of the cell cycle? S phase, right? Because during S phase, DNA polymerase is grabbing onto bases and adding it to the growing DNA strand, right? So cells in S phase can be identified by detecting the incorporation of base analogs such as BRDU. So you have an antibody against BRDU. What we've done is we've used a BRDU, anti-BRDU antibody that's green. And so if the cells are green, then that means they've just gone through what phase of the cell cycle? S phase, because that's when you would put it in. That's when the cell would incorporate the base analog. It's finished S phase. Yeah. Any questions about BRDU? These methods sometimes do confuse people. So if you're confused, you're not alone. Raise your hand. Yes?
So does it inhibit the cell cycle, or it's just a way of
It's
monitoring
just-
where it is?
It's just a way, yeah, so it does not inhibit the cell cycle. It is not an inhibitor, it's a marker. So it's a way, so like normally you wouldn't be able to differentiate between thymidine from last phase, like in the parent strand of DNA and in the daughter strand of DNA. But by having BRD, or there's just a bromine added into the structure of thymidine, it makes it easily detectable by an antibody, and so then you can know what most recently happened. Yes?
So if the
If the green wasn't there, that means it hasn't gone through S phase yet. So all cells here are kind of stained with red. So all of these cells haven't gone through S phase yet. Yeah. Have not gone through S phase yet. Only the green ones. Yeah. They will eventually. It's a timing thing, right? So if you take a picture of your skin and you do this kind of, and let's say you were able to detect which cells went through S phase recently, it wouldn't be all or nothing, because they're on different schedules. Okay, any other questions about BRDU staining? Okay, skip this practice problem because A and B appear to be missing, but it's important to be able to understand it. You'll see it on your thing. Okay, so flow states recently gone through S-phase. That I believe was on the quiz this weekend, right? Okay. Flow cytometry. So flow cytometry, this looks complicated, but I put it in here so you can understand it's a machine that counts single cells. Okay? And so by having the ability to count single cells, you could do things like stain them for different things and count how many cells are positive for that stain. So for example, people use flow cytometry a lot with HIV research. Let's say you go to get your T cells counted because you're an HIV patient, and they can actually count your CD4 positive T cells because you can stain the cells with an antibody that's going to recognize CD4. That's going to have some kind of color, and then you put it through this machine, and it's going to count, like, you tell the machine, count 100 cells. Actually, it's more, count 10,000 cells. And it's going to tell you the percentage of cells that are green or whatever color, and those would be your CD4 positive cells, because you've basically taken an antibody, thrown it on the cells, and said, hey, find all the CD4 positive T cells. Why am I telling you about this? This is not an HIV class. Well, you could also use it for studying cell cycle, because you can stain the DNA and study how much DNA is in the cell. Does the amount of DNA in your cell fluctuate through the cell cycle, or is it always the same? Think about it. Does the amount, the quantity of DNA change throughout the cell cycle? Yes. When? S phase, because it doubles, right? Right? You double your DNA during S phase. After M phase, you're back down to 1 copy. But between S and M, you're doubling, right? So this is useful. So basically what you can do is you can stain your cells with propidium iodide, or PI, and it gives the cell it's like DAPI. It acts just like DAPI except for it's orange. And it's going to bind to the DNA. And then you can run it through the flow cytometer. And you can say to the flow cytometer, count all these cells and plot them on a graph based on how bright the orange is. If the orange is dim, there's less DNA. If the orange is really bright, there's more DNA, right? Because the quantity of DNA and the intensity of the orange correlate. And so if you look at this graph here, this is how it kind of comes out. So this is your number of cells, right? 0 cells, 10,000 cells, or whatever. So, or 0 or 100%. And if you take cells at any given time, stain them with PI, and put them through a flow cytometer, most of your cells will probably be in G1, and they'll have 1 copy of DNA, right? In G1, you have 1 copy of your DNA. Then during S phase, it's going to increase. That intensity is going to increase, but it's a pretty small number of cells at any given time that will be in S phase. So that's why it's so low. But notice it's this kind of broad transition, because over time you're increasing your quantity of DNA, so you're going to increase your intensity of your orange. And then between G2 and M, you'll have completely double your DNA. So that's going to be your brightest orange. And after M, you kind of end up in that 1 pile again, right? So this always confuses people. So I made a practice problem, but quickly before, I just wanted you guys to see exactly what the PI looks like. And this is PI staining. It's this kind of orange color, and you can count it like a histogram, like what we just saw, or you could do like a scatter plot. But still, it's counting numbers and intensity of PI, and the more intense the PI is, the more DNA is. Here, these cells are double stained with PI and with phosphohistone H3, and a phosphorylated histone H3 is associated with chromosome compensation. So that's kind of like a double way to check that you're in mitosis. And as you notice, you're dealing with intensity. So your PI staining intensity goes this way, and your phosphohistone H3 intensity goes this way. And so the things up here, these are your mitotic cells because they've got the phosphorylated histone H3 and they're also more intensely PI stained. Yeah, so there are more cells in G1 phase. But when you're studying something, you're going to treat your cells in some way and see how that changes. So I made a practice problem for you guys to practice it. What time is it? Oh, that's right. Okay, thanks guys. So what I want you guys to do is do this practice problem, and I'll tell you what to do. It's kind of crazy. Some of you have it in front of you. So basically, you're studying a virus. And you think that this virus has some effect on cell cycle, and you want to know what that effect is. And so what you do is you decide you're going to PI stain, and you're going to do flow cytometry, and you're going to see if there's any effect of the virus on cell cycle compared to media alone. So over here, you've got media only. So that's just cells growing normally. This is the normal distribution, G1S, G2M. And then this is media plus a virus. And so I want you guys to take a minute and look at this. And I want you to be able to decide which peaks are what phase of the cell cycle. And tell me, did the virus have any effect on any? Okay, so just take a minute to look at this. Sorry, guys. But it helps if I lifted up the screen. That helps. Yeah.
That's why there's Texas.
Yeah.
I work in the
guys, lots of interesting questions. Okay, so the way to approach this is to start first with your control. And what you need to do is you need to say, okay, so you label it. So what's this peak? G1. What's this? S. And what's this? G2. G2F. Right. And how do you know that? Other than that I told you that. But how do you know that? We're looking at PI intensity, right? And so the more DNA you have in your cell, the more staining you're going to get, right? Does that make sense? Is that logical? Yes. And you are, right? You're going from one copy here, and you're going over here. So you are increasing your activity. Don't look at these peaks. These are cell numbers. So people always look at this as intensity. Intensity is x-axis. Cell numbers are this axis. So this is 0 cells. This is 100% of cells. So G1 at normal, without the virus, in G1, normally you've got about 80% of your cells there, right? With one copy of DNA. During S, it's going to increase. Why are there so few cells in S? Well, just because it's a really short phase, it's a short period of time, so you're not going to have all your cells in S phase unless you synchronize them, which this is not what's happening. And then here's where you have double the DNA. You only have double the DNA at the completion of S phase, right? And so that would be G2M up until cytokinesis. Because once you have cytokinesis, you're back to one copy per cell, right? So this is normal. What's happening here? The opposite, okay. So the opposite, what do you mean? What are you going to study now? What's your hypothesis? What is the virus doing? Yeah. So is it preventing cytokinesis? And so they're all stuck in M. Maybe. That's a hypothesis that you would test. What?
Could be
so maybe it's stuck in G2, preventing entry to mitosis. Maybe. And so then what you would do is you would go into the literature, you would study how this normally happens, and then you would start to look and see, well, what kinds of things could be not working well. Viruses love to bind to proteins and make them go away, or make them do different things for the sake of the virus. Okay, so go ahead and go do what you gotta do. Enjoy your Thanksgiving. Okay, so normally you have a mixed population of cells that are different places of cells. That's just how it is. And so at any given time you'll see cells that are all different places. But if the cells have a mutation that affects cell cycle in such a way that it's going to halt cell cycle somewhere, then it's going to halt, then they're all going to kind of cycle until they reach that roadblock. And this roadblock in this case is G.
Hello. I was wondering, could you write me a recommendation for some locations?
Sure. What are deadlines?
I'm gonna let you know. I'll probably send you a spreadsheet with all the programs and their deadlines.
Okay. Sure. Hi.
You have a few minutes?
Yeah.
Oh, that's great. Oh, I love it. Alright, I'll do
Well,